Looks good.
Okay. Um, so, as I'm sure all of you know, my name is Lillian Peterson, and my project is an activity by contact model to predict enhancer gene connections, which is a tool to increase our understanding of cancer. So first, a little bit of background. Genes are expressed at different levels in every cell, and this is what makes cells different from each other. For example, your skin cell is different from your muscle cell, even though they have exactly the same DNA. Transcription factors are proteins that regulate gene expression by binding to specific sites on the genome. This causes nearby genes to be transcribed into mRNA and translated into proteins, which carry out all the functions in our body. However, there is still a limited understanding of these exact mechanisms. An old theory is that enhancers regulate genes that are nearby on a linear sense of the genome. But a new and emerging theory is that enhancers actually regulate genes that are close by on a more three-dimensional sense, because the genome actually takes on a very particular three-dimensional structure. So my goal was to determine which enhancers regulate which genes using this new theory that enhancers regulate genes that are physically close. I analyzed data from 4 different DNA sequencing methods to determine key information about the genome. So RNA-seq measures the gene expression of every gene in a group of cells. ATAC-seq measures the locations and sizes of enhancers. Hi-C measures the contact frequency between any 2 locations on the genome. And I use Hi-C to determine whether enhancers and genes are actually physically close. And ChIP-seq is just an alternative method to measure the locations and sizes of enhancers, just like ATAC-seq. So I created an activity by contact model to predict which enhancers regulate which genes. The activity by contact between an enhancer and a gene is defined as the activity of the enhancer times the contact frequency between the enhancer and the gene, divided by the sum of that quantity for every enhancer that comes into contact with that gene. And I actually created 2 different versions of this score. In the first version, the activity of the enhancer is defined solely by ATAC sequencing. And in the second version, which I'll refer to as modified ABC, the activity of the enhancer is the geometric mean between ATAC sequencing and ChIP sequencing. And I use this to determine whether ChIP sequencing was necessary in the calculation of the ABC score. And in both versions, the contact between the enhancer and the gene is defined by Hi-C. So basically, the higher the ABC score is between an enhancer and a gene, the more likely it is that that enhancer regulates that gene. So you may be wondering, like, why is it important to know enhancer gene connections, and how does it pertain to cancer? So I'm going to answer those questions for you right here, using this diagram. So in the genome of a normal B cell, there's a gene called EDB6 on chromosome 12, which, like any other gene, is activated by enhancers and then translated into the protein EDB6. EDB6 itself, though, is a transcription factor, meaning that it goes and binds to many other places in the genome, where it causes the activation of other genes that are supposed to be on, so everything is working properly. The gene RONX1 on chromosome 21 functions similarly. But in the EDB6 RONX1 subtype of B-cell leukemia, there is a translocation between these 2 chromosomes right in the middle of these 2 genes. This leads to the formation of a mega mutant transcription factor, EDB6 RONX1, which then goes and binds to many incorrect places on the genome and causes the activation of genes that are not supposed to be on. These genes turn into oncogenes, which are genes that lead to cancer. In this example, we know what the mutant transcription factor is. It's EDB6 RONX1, but this is not usually the case. We usually do not know what mutant transcription factor is causing the activation of these oncogenes. So my goal was to identify these transcription factors, and then we could create drugs that target these mutant transcription factors and thereby treat the cancer. First, I conducted a validation. I tested the model in the K562 cell line using a set of known enhancer gene connections. I obtained raw sequencing data from online databases and wrote 4,200 lines of code in Python, R, and Bash shell script to process hundreds of gigabytes of data and create the ABC score for every possible enhancer gene connection. First, we'll look at the distributions of ABC scores. So here we have the ABC score increasing on a log scale, and on the y-axis we have the frequency. The green shows the distributions of ABC scores for enhancers and genes that are connected. So in these cases, the enhancer does regulate the gene. And in the blue, we have the distribution for enhancers and genes that are not connected. As you can see, connected enhancers and genes have significantly higher ABC scores than those that are not connected. This plot shows the same thing, but for the modified ABC score, and the same trend holds true. Those enhancers and genes that are connected have higher modified ABC scores than those that are not connected. I also compared the ABC model to the old model of linear distance to make sure that the ABC was working better. So here we have the linear distance, which is just the number of nucleotides or base pairs along the genome. No matter like how it curls, it doesn't matter. So we have the linear distance increasing on a log scale, and on the y-axis, again, we have the frequency. And you can see that enhancers are closer linearly to genes that they regulate than genes that they don't regulate. In order to directly compare these three models, I created a precision recall curve. This figure explains precision and recall. So in the green, we have the positive elements, or the enhancers and genes that are connected. And in the red, we have the negative elements, or enhancers and genes that are not connected. And in the model, we have the elements that the model predicted to be true. So precision is defined as, out of all of the selected elements, how many are true? And recall is, out of all of the positive elements, how many did the model select? So a perfect model would have both 100% precision and 100% recall. You can see that the ABC score performs significantly better than both the modified ABC score and the linear distance at predicting enhancer gene connections. So this is important for two reasons. It shows that the ABC score is better than previous methods at predicting these enhancer gene connections. And it also shows that ChIP-seq is not required for an accurate ABC score. So labs that are interested in using this method only have to conduct ATAC sequencing. So next, I applied the model to study 24 B-cell leukemia patients. First, I identified each patient's subtype by conducting principal component analysis of their 1,000 most variable genes and comparing it to about 2,000 previously identified samples. So here you can see how the MCG samples, which are the samples that I worked with, grouped into the different subtypes, and the subtypes are grouped based on the color. And the main subtypes that were represented in the data were hyperdiploid, DUX4, EDV6 RONX1, Philly-like, and PAX5AL. And these are just 5 different subtypes that are pretty common. So these were the subtypes I used throughout the rest of the application to leukemia. Then for each subtype, I identified both differential enhancers and differential genes, and then identified differential enhancers that had high ABC scores between them. So here's a visual representation of what that looks like. In the differential subtype, there is an enhancer that is much more activated than all the other subtypes, and this regulates, and this is nearby to a gene which has a much higher gene expression than the other subtypes. So this gene is likely an oncogene because it is not as expressed in what you might call the normal state. And then if this enhancer has a high ABC score to this oncogene, then it likely regulates that. So if we could then create a drug that targets this enhancer, then that would treat the cancer. So that would be the end goal. So here are 3 interesting examples where this is the case. This plot shows the gene expression of the FOXO4 gene against the ABC score between the FOXO4 gene and this enhancer on chromosome X. You can see that the hyperdiploid subtype has both a much higher gene expression and a much higher ABC score than all the other subtypes. The FOXO4 gene is actually an extremely important gene because it's a known leukemia oncogene that's involved in cell cycle regulation and apoptosis. Here, I identify an enhancer that regulates the FOXO4 oncogene. Next, we could create a drug that targets this enhancer and thereby treat the cancer. Here are 2 other plots that show very similar things, but with the genes HU1 and GATA3, which are also very important genes involved in things such as DNA repair. And again, I identify other possible drug targets to treat other subtypes of leukemia. So in conclusion, the ABC model is a better predictor of enhancer gene connections than previous methods. This model builds a better understanding of the mechanisms of gene regulation and supports the more recent theory that enhancers regulate genes that are physically close. The ABC model can create a better understanding of the activation of oncogenes, identify mutant transcription factors, and lead to the development of new drugs for targeted treatment of cancer. And I would like to thank Dr. Graham McVicker, Dr. Jesse Dixon, and Dr. Jixiao Zhu of the Salk Institute for Biological Sciences for guidance in this project. They directed me to relevant papers, provided data sets, and answered my questions on methodology. But before I go, I got a list of questions from Dr. Einhorn a few weeks ago, so I have a couple of slides devoted to answering some of the questions that he brought up. So, whoops, looks like these were coming in one by one. So the first question was, how does your ABC model compare to the Fulco et al. Paper? So over the summer when I was starting to work on this project, I saw a preprint for the Fulco et al. Paper who proposed the ABC equation. And this paper was actually preprint, and it wasn't published until just December a few months ago. And so Fulco et al. Proposed the ABC equation and tested it on a very small data set, but did not do anything else with it. And so I saw this equation and I thought that the applications for it could be vast, especially in the field of cancer, where understanding enhancer gene connections is so important. So I add on to the research done by Fulco et al. In 3 important ways. First, I test different versions of the ABC model using either ATAC-seq or the geometric mean between ATAC-seq and ChIP-seq, and I find that the one with only using ATAC-seq works better. Second, I validate the ABC model on a much larger data set than they were able to use in theirs, because this data set was only published a few months ago. That's how new all of this research is. And third, I apply the ABC model to study cancer, which was not done at all in Fulco et al. Or has not been done before. The second question was, are open chromatin regions always open? So the answer is that yes, the genome takes on a very specific 3-dimensional structure. And every cell of that type has the exact same folding, the exact same structure. So therefore, the open chromatin regions, which are where the transcription factors bind, remain the same throughout time and between cells of the same type. But a note is that during cell division, the whole genome curls up really tightly and temporarily loses its shape. But after division, both daughter cells regain the same specific folding pattern. So more folding questions were that you are implicitly studying the 3D effects via the contact frequency data. Yes, contact frequency tells how close physically an enhancer is to a gene. So I don't actually have to know exactly how the genome folds. I only care about the contact frequency between two elements at a time. My second question was that I'm not explicitly studying protein folding, and that's true, I'm not. The ABC model is concerned with the contact frequency between an enhancer and a gene, both of which are located on the genome. So I'm not examining any proteins. Where am I getting this data? For the validation, I get RNA-seq and ChIP-seq from ENCODE, which are online databases that have a lot of data on them. And then I get the Hi-C and ATAC sequencing from the Vicker lab at the Salk Institute, which was the lab that I was working with. So they ran a lot of their own wet lab experiments. And for the application to leukemia, I got everything from, again, from the Vicker lab, except I did not use ChIP-seq data. I was also asked to explain principal component analysis. So principal component analysis is basically you have some data set that's in some number of dimensions. So here's an example with 2 dimensions, but I'm actually using something that's more like in 1,000 dimensions, because for each of these samples, I'm looking at 1,000 most variable genes. And then you take the dimension along some axis that has the most variability. And that would be principal component 1 that you can see here. And then you take the next axis that has the second most amount of variability after eliminating the first axis. So that would be principal component 2. So that is how I get this plot for identifying the subtypes for the BALL samples. But there's just a package that I used in R to actually do it and make the plot. And finally, I was asked to go into a little bit more detail about the data processing. So this was a figure I included in my paper, I think, and I can see why it would be sort of confusing. So I made a simplified version on this side. So when I get the data, so this is an example with RNA-seq data because it's the easiest to understand, but the process is very similar with the other types of data. So when I get the data, it comes in FASTQ files, which are basically just compressed versions of like ATT, CC, ATGC, and so on and so forth. So there's a lot of processing to be done. So then I run a lot of code and end up with unnormalized text files. So the number of reads on each gene for each sample. So if a gene has 100 reads, then a sequence of that gene was read 100 times in the sequencer. And sequencing is sort of complicated and it would take a long time for me to go into it. So if you're confused, you can check out this video, which does a very good job at explaining it. And then I have to conduct normalization so that I can compare the samples to each other. So the first type of normalization is RPM, where I divide the reads on one gene by the total reads in the sample. And that's because sometimes you have really deep sequencing and sometimes you have very shallow sequencing. So you want to normalize by the total number of reads that were read. And then I normalize by RPKM, which is I divide the RPM by the length of the gene, because a read is only 200 base pairs long. So longer genes will naturally have more reads aligned to them, even if they are not expressed more. And then the third normalization step is quantile normalization, where the distributions of the RPKMs between the samples should match. So I use some R package to conduct quantile normalization to make sure that they do. And all of these steps are very standard in this field. So then the final product is the quantile normalized RPKM gene expression for each gene and sample, and then you can compare the samples to each other. And that is it. Oh, I had one more note. An RPKM, just to give you some idea, an RPKM of 0.1 is pretty low. We don't use anything below 0.1 because that would mean you can't pick up on differences. An RPKM of 5 is quite high. And I think I'm done.
Thank you very much, Lillian. A great presentation as always. If we were doing this in person, you'd be hearing a round of applause now. That's a little hard for us to do, but just imagine it in your mind. I think you're used to it. Judges, I'll turn it over to you for questions.
So Lillian, this is Dave Janicki. Question about your modified ABC, and you talked about a geometric mean. Can you go into a little bit more detail about why that's been proposed, and is that sort of the only answer, or could there be some other things going on that might be a difference than just a pure geometric mean, to say where this might go and why it doesn't work as proposed, but maybe something else could come in there to balance more of these measurements. Thanks.
Yeah, that's a good question. So I only created 2 versions of the ABC score, partially because the run time was very, and the processing time was very long. And I used a geometric mean because that is what was suggested in Fulco et al. In fact, they only, like the ABC model that they used, only used the geometric mean between ATAC-seq and ChIP-seq. And the exact reason why it's a geometric mean, I'm actually not sure. So I think something that would be very interesting in the future would be to try different, like more different versions of the ABC score using maybe just a straight mean, or just using ChIP sequencing. The reason I didn't create an ABC model with only ChIP sequencing is because the end goal was to apply it to these leukemia samples, and we didn't have any ChIP sequencing data for that. So we really wanted to know, like, can we just use ATAC sequencing data, or like, is ChIP sequencing really necessary?
May I ask a question? I'll follow up. I think it may make sense to think of the ATAC and ChIP as being 2 features of a different nature, and so to combine them, you wouldn't want to use an arithmetic mean because it means adding 2 quantities of a different nature, but you can multiply them, and so geometric mean might make sense in that sense. But my question is, have you made the comparison when you look at leukemia data? What would be the comparison between your results with this method and the results that you would have obtained using the linear distance metric alone, right? Would you have identified the same potential oncogenes and their enhancers?
So I think that if I had used the linear distance model for the application, then it just would not have worked very well. And as you can see, the precision and recall for the linear distance, which is the red line here, is pretty low. So, you probably would have been able to find like 1 or 2 interesting results where it guessed correctly, but I don't think that there would have been as many like interesting finds. And the problem is that there's no validation data set once you get to the leukemia samples. Everything is just sort of a best guess, because as of right now, there is 1 data set that accurately gives connections between enhancers and genes, and that's the Gasparini et al. Data set I was using, and that's only a data set for the K562 cell line. So once you step out of that, you're in sort of completely unknown territory, and every result you come up with as of right now is just a best guess, and it would need more validation before anything would actually be like go into drug treatments. So you really want to use the best model you have, and I wouldn't say that there's even much of a point to testing out a model that doesn't work as well, because there's no validation data set.
Thank you.
Lillian, this is Sharon DeLand, and my question is, Can you talk about which part of this project was the most challenging from a computational standpoint and how that was handled in order to make it more efficient or accurate or something along those lines?
Yeah, so I mean, I would say personally for me, the most difficult aspect is that I had to learn a new language. I've been programming in Python for a very long time, but there are a lot of packages that are only in R that you have to use for a lot of this like biostatistics stuff, because a lot of it gets pretty complicated and there's only like one package to do it and it's in R. So for me, that was a very difficult step to learn a completely new language. But like computationally, it's just a lot of data processing, I guess. And figuring out the data processing steps at the end did take like quite a bit of time, especially to understand just what was going on, because this was a completely new topic for me. I've never touched biology or molecular biology before. So I had to spend a lot of time researching just like what all these different things are and what it means. And for me, that was probably the most difficult thing. I had to spend a long time learning the content in order to understand the model I was building.
Okay, thank you.
Judges, does anyone else have any questions? Stephen?
Yes, I have a simple question. I'm also a newbie in this space. How do you go from the base sequences to genes? Going from FASTQ to then identifying the genes.
Yeah, so what the okay, so you have your sequencer, and it has, so it reads the mRNA data. So basically, when a gene is transcribed, it's transcribed into mRNA. So if a gene has a higher expression, it's transcribed more. So there's going to be more mRNAs just laying around in the cell. So then you take all of the mRNAs within a group of about 10,000 cells, because that's what you need. And then you sequence them in an Illumina sequencer. So meaning you get the sequence on a section of this mRNA every time, like for some portion of the mRNAs that are, total mRNAs are in there. So if you read this mRNA more times, then this gene has more expression. So
well,
then
my question's a little bit more basic. Going, the FASTQ gives you the nucleotides or the base pairs?
yeah, so then I'm getting,
how do you go from that to a gene? How do you find,
so,
which ones define a gene, I guess?
So
or
what
you
you
answering
do
that?
is you have this sequence, which is a read from the mRNA. It's about 200 base pairs long, and then you use a program to align it onto the genome. And then if you align reads onto like this specific spot, like 200 times, then you know that that gene has 200 reads, and then you go through all the sequencing steps, all the processing steps. Does that make sense?
Yes, yes, it does. Thank you.
Okay, I mean, the exact like program I use, if you're interested in that, is BWA mem. So it's a lot of programming in what's it called, Bash shell script, and so you have to mem it, and then you get you have aligned reads.
Perfect. Thank you. Awesome.
Judges?
Is it possible for me to ask one very brief question?
Sure.
So were you an intern at the Salk Institute while starting this project?
Yes, I interned at the Salk Institute over the summer, and then I continued to work on it over the school year. So that was a great experience.
Good. And were you using their computers after you left?
I was like using my computer, but I had to do SSH onto their computer because the DNA sequencing data is confidential and can't be copied off.
Okay. Thank you very much.
Yep.
All right, we're out of time, Lillian. Thank you so much. Great job as always.
Thank you.
Go out and enjoy the rest of your day and take a nap.
Okay.
Very nice, Lily.
Bye.